I want to welcome up Dr. Richard Barker. He's a research scientist with the University of Wisconsin-Madison. And Dr. Barker, I do have to say, you have a very impressive resume. It's very long. I won't read it because it will take up a while. But you have probably the most energy I've met of any astrobotany, space agriculture nerd. So I applaud you, and I'm really excited to hear what you have to say. So with that, the floor is yours, Dr. Barker. Oh, it looks like we might not be getting any audio from you, Dr. Barker. Test, test.
There we go.
Hey, there we go.
Excuse me. Okay, cool. Can you see my presentation?
We cannot.
Can you hear me?
We can hear you though.
Okay, let me just share my screen one more time.
So that,
Well, I just want to say it's a real honor to have the opportunity to share some of the research I've been doing in the Gilroy Lab, and to follow Davy. What an amazing person to follow. Oh golly, the portfolio that they have is really amazing. So the research I've been doing, we presenting today, has been conducted at the UW-Madison. I'd like to support Gravity, the Blue Marble Institute, CoSE, the Capital Science Environment, and NASA GLab for supporting me throughout the years. Along with astrobody.com for helping us share some of the methods we use for the research students around the world. Today, the project I'll be presenting is a combination of two projects: TOAST, Test of Arabidopsis Space Transcriptome, and TikTok, targeting improved cotton-free orbital cultivation. These projects are the brainchild of Professor Simon Gilroy and Dr. Sarah Swanson, and myself and the team at the Gilroy Lab have helped pull these things together. There's so many collaborators that I can't thank them all, but there's a slide at the end dedicated to them. So ultimately, what I'm going to try and describe to you is how we have used fundamental plant biology and machine learning, and then translate this information into improved crop growth in microgravity. Okay, so what you see before you are plants going on the International Space Station, and then plants going on Earth. Can you tell which ones have gravity and which ones don't? These particular plants were growing in the veggie growth system on the International Space Station, so they had light. We were able to get these samples back to Earth, and then we were able to sequence the messenger RNA in the roots and shoots in order to provide some kind of insights into the genetic response that these plants are experiencing. These data, along with the analysis of the physiological patterns that you can see before you, are now being prepared in a manuscript that will be published shortly. As these data, the raw data, will be available in NASA GLab, and also available through TOAST. TOAST is an interactive relational database that we've built. It's available on the Gilroy Lab website, which binds together the multi-omics data that's been provided by the space biology research community as part of the NASA GLab collaboration. So essentially, we've integrated transcriptomics, proteomics, epigenetics data into a multi-omics data explorer to help researchers and students around the world find biomarkers of interest that may help them conduct experiments to further space agriculture. We have recently published a paper. It just came out in Nature Microgravity just a couple of weeks ago, describing the meta-analysis and the insights that come from the first 15 spaceflight transcriptomes. The wonderful thing about using a transcriptome is you have over 24,000 different biomarkers to assess how an organism is responding to its environment. So let's just take our model organism of Arabidopsis thaliana. Here you can see Scott Tingle loading the samples into the veggie growth system. These particular plants are really, really sensitive to light and gravity and all those other stimuli that help them orientate themselves traditionally on Earth. So by extracting the RNA and sequencing it, we can get insights as to how these plants are perceiving the environment and any potential stresses that might exist within the veggie growth system on the International Space Station. In order to extract information, we took a machine learning approach. So we a stress pattern recognition system that used a autoencoder to take data from GEO and essentially make a pattern recognition system, a stress recognition system, a bit like the coal miners used to do with canaries back in the day. So essentially, instead of having a canary in the coal mine, we're saying Arabidopsis can provide a lot more insights that can allow you to change the environment before the unthinkable happens. So essentially, this multi-stress pattern recognition system takes 85 different transcriptional studies from the GEO repository and creates a multi-pattern recognition system that can recognize different types of stress, be it drought, light, heat, stress, all the different kind of major groups can be decoded by analyzing their transcriptome, the plant transcriptome, through this software. So when we put the Apex-5 data through and we're able to look at the roots and the shoots, the hypothesis we had was actually based on some of the earlier work done by Marshall Portersfield and some of the people that presented earlier in this conference, that essentially we believe that plants would be experiencing hypoxia stress in spaceflight due to the lack of convective mixing of gases. The color coding you can see here, more red means more similar, and more blue means more different. So although there is some similarity between hypoxia and spaceflight, ultimately, when you focus in just on the roots, what we can see is that the major stress that the plant roots are experiencing in the veggie growth system appears to be more closely related to a low pH stress or salt stress. So let's think about hypoxia, the initial hypothesis that we had, and essentially the respiration process that's occurring in the mitochondria may not have enough oxygen due to the lack of mixing of gases in microgravity. So rather than producing CoA and the citric acid cycle performing normal respiration, instead we have more products going down into the lactic acid pathway, leading to the production of, well, an acid, right? Low pH stress. So in conclusion, our initial research into Arabidopsis, doing some fundamental biology research, led us to believe that acidosis is being experienced during plants in the veggie growth system in orbit as a result of the microgravity environment. So we can provide recommendations to the hardware producers. In other words, we recommend that there should be more regular monitoring of the atmospheric composition when plants are grown in microgravity environments. In addition, people should take note of the rhizosphere pH, and there may be opportunity to reduce the stress associated with microgravity by changing the pH in certain areas of the root system. Ultimately, to further build on this data, we need more time series data, and we'll be providing this advice to other NASA researchers over the coming years. We believe there's a lot of new insights that can be made available through the use of microgravity simulators and atmospheric chambers on Earth. So using this data we had gathered from Arabidopsis, we really wanted to find a way to apply this in a crop that would make space agriculture more sustainable and more reliable moving forwards. We were quite lucky at this time because there was a call from proposals from CoSE, that's the Center for Advancing Science in Space, that was sponsored by Target, in which they wanted to design some experiments that could be performed on the International Space Station that would lead to increased cotton sustainability moving forwards. This is a great example of corporate social responsibility in action. So to put it in perspective, cotton is a really thirsty crop. 1 kilogram of cotton uses over 10,000 liters of water every day. So all of those dollars in your pockets, those shirts on your back, all of us have benefited from this cheap source of cotton that the world has had. However, over the last 30 years, time-lapse photography from the International Space Station has revealed that this inland sea in Uzbekistan has almost disappeared as a result of, well, very efficient irrigation in the former Soviet Union's state of Uzbekistan. For those of you that aren't familiar with the Aral Sea, its size is about the size of 3 Greater Houstons. So that's a really big sea that's disappearing because of irrigation. So maybe there were alternative strategies that could have been applied that would have allowed us to continue to have this large inland sea. So I just want to highlight this study from Zhang et al., in which they had wild-type cotton plants growing in these dry fields. They had a genetically modified version where they overexpressed AVP1. For those of you that aren't familiar with AVP1, it's an Arabidopsis vacuolar pyrophosphatase. So essentially, it's a proton pump that sits in the vacuole and moves protons into the vacuole, making it more acidic. The consequences of this is a larger plant, which is more resistant to drought and salinity in the field. It could be an alternative way to maintain yields in these more stressful future agroecosystems on Earth. But remember, when we looked at the Arabidopsis in this spaceflight environments, we realized its roots were experiencing acidosis. So maybe that lactic acid is producing protons that are being harmful to the plant. So what if we were able to transfer them into the vacuole? And how could this affect plants in the spaceflight environment? Well, these were the questions that we wanted to ask. And so we applied to CoSE and to NASA and had the opportunity to go to the Kennedy Space Center to design a new method to grow these plants, so that we could not just grow wild-type plants, but 2 genetically modified versions that had overexpressions of this AVP transgene. The astronauts grew the plants and photographed them at time points online, and the samples were frozen and returned to Earth for processing, so we could do molecular analysis on the biochemistry and the RNA. Just looking at the photos for now, we were not able to extract pigment. Pigment is a really powerful indicator of stress. But as you can see from this photography, the ambient light environment was not conducive to being able to extract reliable patterns from the spectrum of the photos. However, we were able to extract morphological data related to the root system architecture. You can see on the right here, this time series data, we can see the wild type root getting deeper and larger over time. But what we can also see is the two AVP overexpression lines, their roots grew faster and deeper in the spaceflight environment. You can even note the moments when the lateral roots emerge earlier as part of this feature. So essentially, when we look at the final time point, we can see these transgenic lines have got deeper roots, and this results in the larger shoots that are more stress resistant, which has been previously demonstrated in the field. So in summary, the AVP overexpression lines grew better in flight relative to wild-type controls, and that's in part due to the earlier lateral root growth. So we mustn't neglect the hidden half of biology. There's a lot of really fascinating insights that we can gain from looking at roots during these early stages of seedling development. And there's a lot more cellular and molecular insights that are coming from the RNA-seq. Because of the short time frame, I don't have the capacity to delve into the changes that we're observing in the subcellular machinery and cell walls, but happy to discuss that with you at any point in the future. So again, in summary, with the RNA-seq, the really interesting thing was that functionally, it really did resemble the wild type of Arabidopter's response to spaceflight. But there are further insights available in the near future related to the distinct characteristics of the CoSE genome, such as its polyploidity. So the questions for this community moving forwards: could this technology be applied to underutilized crop species that might have some really new novel benefit on Earth or beyond? And can this technology be combined with green revolution alleles? Because remember, bigger plants aren't always better. And so maybe there's an optimum balance that we can engineer in the future. I really do want to thank all the awesome people who come regularly to the NASA Gene Lab Analysis Working Group. It's a group of collaborative scientists that meet up every couple of weeks to discuss questions about space biology with the goal of trying to help increase biodiversity at home and beyond. So with that, I've rushed through. I've only kind of skimmed over the surface of some of the science that we've been doing here. And so if you'd like to find out more, please do come to NASA's working groups and we can continue the conversations that this presentation might have started in your mind. And with that, I probably should have got set a timer, but I do know that this conference is running behind, so I did try to keep to time.